Okay, we've been studying cells, and the next area that we need to look at is how do materials get in and out of the cell through the cell membrane. What kind of materials? Well, think about it. A cell has to have food particles, food molecules, and they have to be small enough in order to go. That's the digestion part. Breaks the food down into smaller molecules so they can go in and out of a cell. They need water. They need sometimes other things. They make things, such as one example could be some cells that the islands of Langerhans, they produce insulin. So that insulin needs to get out of those cells and out of the pancreas and into the blood. And they also need to get rid of their waste. So how does this occur? Well, the key concepts to remember as we go over this lesson is how do materials enter and leave cells, and how does a cell size affect the transport of the materials. The vocabulary words that you want to look for is passive and active transport, diffusion and osmosis, facilitated diffusion, endocytosis, and exocytosis. So those are things you want to be familiar with by the end of this presentation. Unit membrane. We've talked about the cell membrane, we've talked about other membranes, and that's, in general, all of these are unit membranes. If it's found outside the cell, surrounding the cell, we call it a cell membrane. Another example of where unit membranes may be found, they can be around organelles, like around the mitochondrion. They have two. They have the smooth one on the outside, and inside they have the one that makes a bunch of folds. Those folds are called cristae. And then we have like the ribosomes, we have the endoplasmic reticulum. So we've got a lot of structures that are surrounded by a unit membrane. And all of those structures, sometimes, not just in and out of the cell, but sometimes substances have to move in and out of the ER, in and out of the Golgi body. What this particular membrane, a unit membrane, has is this very special property of being selectively permeable. The word permeable means that substances will just flow through it. Impermeable means substances will not. Selectively permeable, also used to be known as semipermeable, means that it's choosy. Some substances, some molecules will be able to go through the membrane and some will not. Does that mean it's uniform for all cells of every organism? No. It's not like all the same cell membranes are going to be selectively permeable to every single type of molecule that's the same. One that does, or actually a couple that do, are smaller molecules. Smaller molecules like water, carbon dioxide, and oxygen. Those will easily pass through almost all. But with the other materials, it depends upon the type of cell. Because just like the nucleus, the DNA can't move out of the nucleus. It can't move through that nuclear membrane, which is a unit membrane. So not all membranes are permeable to the same molecules. How does a substance move through a unit membrane? Well, it's one of three processes: passive transport, active transport, and facilitated diffusion. Passive transport and active, the way that they differ is one requires energy in order for the substance to move, the other does not. So if you think of passive and you look at active, which one would you think would require energy? Of course, the active. Think active, you're doing stuff, you need energy. So passive transport is the passage of substances, of molecules, across a unit membrane without having to use energy. And then active would be with. Now if you think about it, what process would be used most often by cells? Would it be passively or actively? Would it have to use oxygen? Have to use energy or not use energy? Well, praise the Lord, it's without. Because imagine, you got trillions of cells all needing oxygen, all needing to get rid of their cell waste, needing food particles and so forth. And that's going on all day long, in and out through our cells. And if we had to expend energy every time something moved across that membrane, I don't think you could eat enough to keep up with the energy needs that all these cells would have. So small molecules such as oxygen, carbon dioxide, they pass through the membrane via passive. No problem. Small, they can move through easily. Passive transport depends on the amount of the substances on each side of the membrane. And that we're going to look at is what causes then the rate of diffusion. Two main forms of passive transport, diffusion and osmosis. We're going to look at diffusion first. Diffusion is the movement of molecules from an area of higher concentration to an area of lower concentration. And it's like if you're going to put sugar in a drink. Okay? First of all, the drink doesn't have any sugar in it. So when you put the sugar in, it's going to start to diffuse. And it's going to move throughout, let's say, tea. Now to help speed that up, you usually take a spoon and you stir it and you stir it and you stir it, which is going to help to speed up the rate of diffusion. Well, diffusion, you can see, means to scatter or to pour out. Well, what's going to stop that? Well, we're going to learn. First of all, you need to know that there's actually stuff that can speed up the rate of diffusion. One we just mentioned, when you stir it. So that's movement. When you stir that drink, it's going to help the sugar dissolve more quickly. Now, let's think about making tea. If you're going to make tea and you like sugar in your tea, are you going to heat it up, put the tea bags, heat the water up, put the tea bags in, and then cool it off in the refrigerator, then add your sugar? No, because it's going to take a lot longer for that sugar to diffuse. You're going to add the sugar right after your tea is ready. You're going to put it right in, stir it. That's going to make it. The heat is actually going to make it to diffuse more quickly, and then if you stir it, that's going to make it. So you have two things. Another thing that will also speed up diffusion is pressure. Now diffusion, I said, it's eventually going to come to an end. It can't keep going on forever because you're going to run out of molecules. When does it stop? When there's an equal number of molecules on either side of the membrane. Now that doesn't mean an equal ratio, like of water to dye. It means of dye to dye. So if you look on the left-hand side of the membrane, they've put a drop of dye, red dye. And on the right-hand side of that membrane, there is no dye. So what's going to happen? There's going to be a higher concentration of dye molecules on the left than on the right. So because of that, the molecules are going to diffuse through the membrane and over to the right. But not all of them. They're going to diffuse until you looking at the red molecules and look at the red molecules on the left and the right after 30 minutes. They're going to be approximately equal. That's when it stops. And it's I like to liken this to riding a bicycle. And like if you're going from an area of higher concentration to an area of lower concentration, which is diffusion, which is a type of passive transport, you don't have to expend energy going downhill. The steeper the hill, the faster you go. Now once that hill ends and it levels out, if you don't eventually start pedaling, you're going to come to a stop. And that's what happens to these molecules. They will stop when they reach an equilibrium. Osmosis is a special kind of diffusion. It's the diffusion of one substance only, and that's water. So it's the diffusion of water through a membrane. Semipermeable cell membranes allow water to pass through them until equilibrium occurs. So yes, diffusion will occur, osmosis will occur until we reach an equilibrium, and that's what stops it. Now facilitated diffusion occurs when molecules pass through a cell membrane using special proteins. They're called transport proteins, and if you look down there, there's two types of transport proteins. There's carrier and channel. The carrier proteins are transport proteins that have to carry large molecules through the cell membrane, whereas the channel proteins are transport proteins that form a pore a hole, and the molecule is going to be small enough then to just flow right through that little hole. Here's a diagram on the left showing you the two different types, and the inside and the outside of the cell. And you can see the channel protein, which produces the little hole, that is in blue. And then you have the carrier ones, which actually change shape to allow that bigger molecule. Now, unfortunately, they didn't make the molecule look bigger on the diagram, but it should. It's a larger molecule. I think it would have been better if they showed the blue going through the blue.
There are two main types of transport proteins: channel proteins and carrier proteins. Channel proteins are tube-like and provide openings through which small, dissolved particles, especially ions, can diffuse. In general, each type of carrier protein has a shape that fits a specific molecule or ion. When the proper molecule binds with the protein, it changes shape and moves the molecule across the membrane.
Active transport, as I said, requires energy. So it's the movement of substances through a cell membrane only by using cell energy. Now, why would it need to expend energy? Well, there's a couple of reasons. Sometimes it's because of the size of the molecule. It's so big, it's, like they got to tug and tug and tug to get that molecule across. But sometimes it's because it's going against the gradient. Going against the gradient, if we look at the analogy of a bicycle, would be going up the hill. And if you're going up the hill, you definitely have to expend energy. You have to pedal because you're going against the gradient. And the steeper the hill, the more energy that you would have to use. Active transport, it shows you here again some of the size of the molecules or going against the gradient. If you look at the diagram there on the left, it says minerals, some sugars, and most amino acids move against a concentration gradient with an input of energy. So if you look at, let's say, the red molecules, this is on the outside of the cell, whereas inside you have 4 already. But they still need that molecule. Let's say it's sugar. So they have got to get that sugar to move into the cell even though there's more sugar in the cell than outside. So that's going against the flow. That's going against the gradient. And the ATP is the energy that is used. Substances moving by active transport can move from areas of lower concentration to areas of higher concentration. Cells can take in needed nutrients from the environment through carrier proteins by using active transport. Another good example would be like a plant that you're growing in a pot. If you're growing in a pot, they need certain minerals. So let's say they need potassium. So they're going to take in, absorb that potassium with the water into the root cells. But eventually they're going to have more potassium in the root cells than left in the soil because they keep taking it out, keep taking it out. Well, they still need the potassium. So what do they have to do? They still need to get that potassium to come into their cells even though it's a lower concentration outside than the higher concentration inside. So they have to go against the gradient. So they would have to expend energy in order to move.
Some carrier protein molecules are used in active transport. They pick up ions or molecules from near the cell membrane, carry them across the membrane, and release them on the other side. Note that energy is needed for this process.
Now, in active transport, they have two kinds dealing with the cell. And if you look at the terms endocytosis and exocytosis, of course, cytosis hopefully will remind you of the cell. Cytoplasm, cytology, cytosis. Endo and exo. Endo, in, exo, out. So endocytosis is when the cell needs to take in substances by using energy. Okay, it's the process during which a cell takes in a substance by surrounding it with its cell membrane. Exo is the process during which the cell's vesicles release their contents outside the cell. Now the contents might be, like we mentioned, insulin, a hormone. So that vesicle would come in contact, it would be moving in the cytoplasm, come in contact with the cell membrane, and that would open up and release the contents, also waste. And then to eat or to drink, that cell has to surround the food molecule, surround the water molecule, and then pinches off and forms a vacuole with those things in it. Now when the endocytosis, when it is an individual cell that is an organism, we call those, remember, unicellular organisms. When it eats and drinks, gets water and gets the food, they actually have other names. It's still endocytosis. They call it, for cell drinking, they call it pinocytosis, and for cell eating, they call it phagocytosis. So you might see those terms as well. And there's still the active transport, the endocytosis. Here's a diagram showing you that both. On the left there, where you have that blue molecule, that is coming in, so the cell membrane is pinching it off, and then you can see it becomes a food vacuole. The other is already in a vesicle, and it's coming and it's going to touch that membrane, and then it's going to release those, let's say those are waste products in there. So how do materials enter and leave cells? The area of the cell membrane must be large compared to its volume, so that substances can move in and out of the cell. Now we're going to look at the cell membrane is what we call surface area compared to the volume. The area of the cell membrane is the cell's surface area. And that would be, okay, let's say you're sitting in a room. You look at the room, you've got 4 walls, you've got a ceiling, and you've got a floor. That is the surface area. The volume would be the air that is in that room. That would be volume. So the area of the cell membrane is the surface area. The volume is the amount of space inside the cell. Basically where all the cytoplasm is and the nucleus and all those organelles.